The Agenda with Steve Paikin is made possible through generous philanthropic contributions from viewers like you. Thank you for supporting TVO's journalism.
COVID-19 was so dangerous when it arrived because it was brand new to our immune systems. Tonight, what we've learned 3 years on about whether infection strengthens or actually weakens our ability to fight off reinfection and other illnesses. Also, what COVID leaves behind in the lungs of those who've recovered. It's Monday, November 14th, and that's next on the agenda. Millions of Canadians have now had and recovered from COVID. A surprising number have had it more than once. That's raised questions around earlier hopes that infection would bolster immunity. In fact, there's currently a pretty vigorous debate about what having COVID does to our immune system, broadly speaking. With us now on that, in Baltimore, Maryland, T-cell immunologist Anthony Leonardi, who is an honorary research fellow at Kingston University in London, UK, currently also a public health student at Johns Hopkins Bloomberg School of Public Health. In Quebec City, Don Bodish, immunologist and professor in the Department of Medicine at Hamilton's McMaster University. Here in Ontario's capital city, Dr. Alison McGeer, medical microbiologist and infectious disease specialist at Mount Sinai Hospital. Colin Furness, infectious control and epidemiologist and assistant professor at the University of Toronto's Faculty of Information. Welcome to you all. All right, I want to know where we all stand on this, and I'm going to come to you, Anthony, first. Are COVID infections impairing our immune systems even after we recover?
Well, recovery is a loose definition there. So if you actually look at certain markers in the blood, there are changes that are long-lasting. So I would argue that it's not full recovery as of yet. But I'm open to hear what everyone else thinks.
All right, and then Dawn, I'll get your take.
Yeah, I think there's a lot of heterogeneity in the responses. So we can think around long COVID as being the extreme example where people have these long-lasting challenges, and some of those are due to changes in their immune response. But where the question I think you're actually asking is for what about the people who just had a reasonable infection? What does their future look like? And data, including some of our own, does suggest that some people do have some lasting changes to their immune response, while epidemiological data suggests that people who've had COVID may be seeking more medical care, maybe ending up going back to the hospital for things that seem unrelated. So I think those two pieces of data together suggest that, at least in some people, there are lasting changes that may contribute to this requirement for more medical care.
Allison.
What we know from previous pandemics is there are likely a group of people who've had COVID who will be significantly affected for at least a long period of time. The perhaps more relevant question is, what about all the people who've had mild infections in Omicron? And that's very difficult to tease out. As Dawn has just pointed out, people who've been hospitalized may have ongoing issues, but the truth is people who've been hospitalized for any infection have ongoing issues. So the challenge here is teasing out what might be something from COVID and what might be something that's just having a viral infection, any viral infection, which changes your immune system, and what clinical impact that has. What we don't have at the moment is strong evidence that for most of the people who've had mild COVID infections, not been hospitalized, that there is an ongoing impact that significantly impairs their response to other infections. Doesn't mean it's not true, just means that we don't have evidence at the moment that it is. And our entire history of, meeting pathogens and dealing with them suggests, I think, that it's unlikely that there's going to be a long-term impact that is clinically significant for the majority of people.
All right, and then Colin, I'll get your take as well.
Sure. I think when we try and answer these questions, we usually try and answer them for everybody. Is there an immune effect on everybody? And I think the better question is and I think Dr. Bowder said it, is there a bad effect in some people? We have some data, such as children being on ventilators for RSV, way above and beyond what we'd expect, despite the fact that RSV has lower prevalence right now than we would expect. So that's a head scratcher, and it suggests that maybe some kids are having a really bad immune suppression problem. It's tricky, but that's what that would immune harm would help to explain some of these observations.
Alison, I want to come to you as well, because I know there's some research out there, just like what Colin was talking about in children. Can you tease that out a little bit more? What are we seeing in terms of what we're seeing in the effects in children?
So I'm just going to frankly disagree with Colin. Okay, I think our problem with RSV is really specific. All kids get infected with RSV in the first 3 years of life. So there's a 30 or 40% infection rate before you're 1, and another similar rate before you're 2, and then some more. And all of us have repeated RSV infections. So usually the first infection is the most severe, but not always. It's a bit complicated. And what happened during the pandemic was we had no RSV activity in Canada, with the exception of Quebec, okay? Quebec is always the exception, who had some activity last year. And so we now have this cohort of children aged 1 to 4 who have not been exposed to RSV significantly and have not had infections, and we're catching up. And unfortunately, when that happens, you catch up all at once, okay? So the 3 times as many people 3 times as many kids in the hospital with RSV infections is exactly what we would expect epidemiologically. It's not about, they've had COVID. It's not about anything except that they're being exposed to RSV when they haven't been before, and a small number of them are getting really sick. And so our challenge at the moment is not actually these kids are getting RSV, but that these kids are getting RSV all at once. None of this takes away from, a lot of important questions about what happens to our immunity and how things work in the long term. But I don't think that the issues we have with pediatric hospitalizations at the moment require an immunologic explanation. There's a perfectly good epidemiologic explanation for what's going on, in my view.
All right, let's return back to the immune system here. Dawn, I want to bring you in. According to the research we have now, which is clear that maybe there's not that much, but for how long after recovery can immune system impairment last?
Well, we don't have very good data, first of all. And I want to make it perfectly clear, we have no pediatric data. No pediatric data. It's very difficult to do those sorts of studies in kids. People don't want to give blood from their kids, and you need a significant amount of it to do those kind of measurements. So every piece of information I'm going to give you is going to be in the adult or the older adult population. And this is a really important distinction, because we have to be clear that just as children aren't small adults, their immune systems aren't necessarily interpretable in the context of what we know about adults and older adults. So with that caveat, I would say we can use things like long COVID as the extreme example, where we know about 1 in 3 people who have long COVID have lasting immune impairment for quite some time. In my own studies of older adults, we can see that people who've had COVID sometimes have some of these immune changes that last over a year. But I think Alison brought up a really important point. We have to be clear that just because we can see a change doesn't mean we totally understand the long-term clinical significance of it. There are viral infections we get in everyday life. For example, there's one called CMV that most of us will have, profoundly changes our immune system, but has very little impact on our long-term health. So although I am cautious, I think the data is unequivocal that we need to avoid COVID. In some people, there will certainly be long-term health problems. I also want to be forthright, because I think it's important that we not overstate this until we have better data.
All right, Anthony, I'm going to bring you into this. If COVID infections are wearing down our immunity, does that mean people will be sick more often? What are essentially the factors and the risks here?
Well, that's a strong possibility. And I'd like to bring up that there are clear, as the doctor said, a lot of these studies were in adults, so we can't, unfortunately, directly extrapolate to children. But there are lasting changes to the immune system. For example, in mild infection, the adults with mild infection at 7 months out they still have a very low level of plasmacytoid dendritic cells. And these dendritic cells are really important for viruses and releasing what's called interferon alpha. Interferon alpha is a cytokine that actually, in early stages of infection, even before infection, can reduce severity of RSV or even prevent infection. So there are strong changes to the immune system that could result in easier infection occurring.
Do we have any data, and I'm going to stick with you, Anthony, in terms of the various variants and how they have an impact on our immune system? Do we know the difference between Delta versus Omicron?
Unfortunately, we had to acquire this data in animals. So again, about the extrapolation, but it looked like Delta was really the worst in terms of pathogenicity. So Delta really harmed the thymi or the thymuses of the mice, and that's an immune organ that helps develop T cells. And it was really just caused a ton of inflammation. With Omicron, it had less of those Delta-like features and harm to the thymus. But with time, Omicron has re-evolved to go back to those changes that were in Delta. So it looks like Delta, so far was really the worst one, but Omicron is returning to a Delta-like phenotype, and that means just the way it is.
All right, Don, I do want to bring you back in. You did talk about long COVID a little bit. Is immune suppression post-COVID different from having long COVID?
Yes, long COVID's a constellation. Many different things can happen. So some people who have long COVID have very clear autoimmunity. So in the context of that infection, their immune system got the wrong message and started attacking their own body. Some people don't have as much evidence of that autoimmunity, but they seem to have damage to some of their organs, at least some of which is probably immune mediated. So it's a real big constellation and shows us how big the variation in what this does. And it depends on how severely sick you are, probably sex plays a role, your age, what variant you got, et cetera, et cetera. So again, we can use long COVID as the extreme of that, both heterogeneity and immune effects. And for many of the older adults in our study, we don't see the very, very extreme immune effects. We see something sort of in the middle. So what's going to be important for us to tease out is how does this impact the rest of their aging trajectory? What other diseases they get, what other infections they'll get.
All right, so we're seeing unprecedented capacity crises in pediatric hospitals and emergency departments. A debate is unfolding across this province. Some experts explain the surge in viral infections as a result of immune suppression post-COVID. Others say immunity debt is to blame. Allison, I'm hoping you can help clear this up a little bit and help explain a little here. Has the idea of immunity debt been misunderstood, and can you help explain what that actually means?
Well, I'm not actually sure what it means when people talk about immunity debt. It's not a term I would use. Really, a piece of this issue, at least, is the fact that we didn't expose children to RSV for a full two winter seasons during the pandemic. So usually in temperate climates in Canada, in the winter season, you get RSV. It starts in October, it goes up to a peak in November, it's stable over the winter, starts to come down in March and go back in April again, and it's pretty reliable. But we've had 2 years with no RSV activity. So there's no question that we now have a cohort of susceptible children who, of ages that pre-pandemic would not be susceptible. It's actually been pretty quiet in pediatric ICUs, with the exception of COVID activity for the last 2 years. And that group of children is now getting RSV infection for the first time. And in Canada, we just don't have much spare capacity in any of our ICUs, but particularly in our pediatric ICUs. So the increase in the number of kids who require hospitalization, just because of the increase in the number of kids who are being exposed to RSV for the first time, is incredibly difficult for our system to deal with. You're not hearing so much about it in other countries because other countries don't have this really, really tight number of intensive care unit beds that we have in Canada. But the there's no question in my mind that the biggest impact of this excess in illness in kids and in our ICUs is related to the fact that kids just weren't exposed to RSV. And in the end, all children are going to get exposed to RSV. The fact that we didn't expose them to RSV during the pandemic is a little bit neither here nor there. The problem is not so much that they're getting RSV, although I will say there's a whole bunch of new RSV vaccines that are going to be on the market for kids in about 2 years. And when we get those vaccines, we won't have this illness and it will be awesome. Okay, but we're not quite there yet. So in the meantime, we can't prevent kids from getting RSV. And our problem is not so much that these kids are getting RSV, it's that these kids are getting RSV all at the same time, which is incredibly difficult for their management in our pediatric ICUs.
Colin, I'm going to bring you in here. Is there any validity, and I'm going to, for the sake of this conversation, call it maybe immunity gap theory, opposed to the immunity debt, but is there any validity to this and to what Alison is saying?
Well, I agree with Alison, and I've learned when I disagree with her, I'm usually wrong. But I'll depart on it a little bit because I think both are going on. There's no question that there's this double cohort effect. That's true. But we did have a spike in RSV in the spring, and we have very low levels now. So it seems to me that if there's not much RSV yet, all this exposure, all this double exposure has yet to happen. And I still don't think it quite explains the number of kids who are seriously ill. This is a testable hypothesis. If it is a double cohort effect, this is going to fade sooner rather than later. If it's immune harm, I think it's going to get worse, and we'll see kids with multiple viral infections and fungal infections too. So I think in the coming months, we'll get a better picture, and I'd love to be wrong.
All right, Dawn, I'll get your take as well.
I think this really highlights a problem with us getting data in a real-time manner in Canada. The perfect way to answer this question is if we had databases where we had records of which kids had COVID and which didn't. And we would expect that the severity would be increased in RSV if COVID was the cause. And we would expect no relationship if COVID was not the cause. But we've got two problems. One, we don't have really good databases where we can get these kind of data in real time. And two, we stopped testing. So even if we could do a database search, we don't actually know which kids had COVID or not. And I actually think I think both things are probably true. I think we're seeing a double cohort effect, and we are maybe seeing some effects of some post-COVID effects potentially. But I don't know where the balance is. It could be more of one and it could be more of the other. And I really want to make a plea for designing better health systems so we can answer these questions in real time, because it really does affect which strategy we would take.
Anthony, I'm hoping
we're on these issues. Can I just interrupt for a moment?
Sure.
I think one of the other really difficult things to interpret is that both COVID and RSV are diseases that are substantially determined by social determinants of health. So that our kids who live in poverty, whose parents are racialized, whose parents are new immigrants, have higher risks of both of them. So there's a really important, the inequity that occurs in our society is an important driver of severe RSV and severe COVID in kids. And so it's partly about having health systems, but it's also about trying to tease out that compounding. Makes it really difficult to know for sure.
All right, Anthony, I want to get your take in there. Is it possible that both ideas can be true, or are we leaning on one or the other?
I personally think it's both. It's definitely true that the mitigations from COVID, like social distancing and mask wearing, reduce the prevalence of RSV. That's, obvious. The question is, could there be an additional effect of immune harm, from COVID? And the evidence is there. Unfortunately, it's not well studied in children, but we see that there are long-lasting, up to 7 months, deficits in dendritic cells. And these cells are they're very much responsible for helping with viral infections, especially in the upper airway. It's well characterized for RSV. Actually, they found that infants and children with severe RSV were more likely to have relatively lower interferon alpha levels. It was a marker for potential harm.
All right, I want to pull up a tweet by Dr. Mustafa Hirji, the acting medical officer of health for Niagara Region, in reference to COVID-induced immune dysregulation having an impact across the province. He writes, "I'd like to see evidence linked to the actual situation at hand. Until then, it is just a hypothesis, but the currently favored hypothesis, so-called immunity debt, lacks both historical and situational evidence and is inconsistent with surveillance data." Doesn't seem like he's any different from what we're saying here, but Alison, what's your response to that?
So I do think that I'm not sure that he and Colin are correct about the surveillance data. There's a couple of issues with surveillance data. First of all, our surveillance data is 2 weeks out of date always, right? It just takes that much time to collect it and post it and get it forward. And if you look at RSV activity now, RSV activity is well above seasonal norms, okay? So it's not true that we're seeing this excess of hospitalization in the absence of RSV. The second problem with, really, Colin, I promise you, I'll send you the data. The second problem is that it's really hard to interpret levels of different viruses at the moment. So pre-COVID, you know, we don't test everybody who gets sick. We don't actually know what the rates are. We test kids differentially over adults. The testing is all done by physicians for who knows what reason. Okay, it's not like we track it. But the advantage is that it's stable over time. So you can look at influenza activity and you can say, okay, 5% of tests, that's the threshold for the season. Now we know we're into the season. And even if I don't really know why people are getting tested or who's getting tested or what's going on, because it's stable over time, it's okay. But our viral testing is not stable now, right? We're doing COVID testing here and other testing there, and everything in the viral testing world has been turned upside down. So now interpreting what seasonal activity means for either Colin or I, frankly, to say, yes, there's lots of RSV activity, or no, there's not as much as usual, really, we don't know. You know, so that just makes it even harder to try to parse this, you know, and you hear the range of opinions. I don't see anything in what's going on that I think is inconsistent with, it was actually not a double cohort, it's a triple cohort effect. But, you know, you're hearing a range of opinions, and this is what science is like, right? You see different possibilities, and then you need to focus on getting the work done to figure out what is true.
Colin, I am going to have to get you to respond to that, of course, because, you know, Alison's saying her name.
Because I've been twitching. No, because I've been twitching. So, you know, I'll believe, Alison, that the recent data shows RSV going up. It should. But those kids who are intubated right now, they were exposed weeks ago when we do have surveillance data. So if RSV is going way up, now we're going to have to see, is there going to be a continued increase in ICU uptake among kids? And if it settles down soon, then it'll be a cohort effect that came and went. And if it continues to get worse, because we don't know how many kids have had COVID at this point, but I think it's most. Anecdotally, it looks like a lot of absences and a lot of COVID. Either it's going to get much worse or it isn't, and I think that's going to give us a pretty unambiguous epidemiological signal. Unfortunately, we're going to have to wait for that, and that's what's concerning.
Knowing the age distribution of those kids will also help, because, you know, if this is a cohort effect, it's going to be kids of a particular age. If this is a COVID effect, it's going to be older kids, probably predominantly, and that will be helpful when we get that data.
And the fact that kids aren't vaccinated below the age of 2 matters as well. If COVID is able to cause more harm in unvaccinated, small, immune immature bodies, yeah, it's upsetting, frankly, to see this happening.
All right, I'm going to shift gears a little bit. We're going to talk a little biology here. To understand why COVID might be suppressing our immune systems, we have to understand what T cells are and what they do. So T cells are a type of white blood cells that defend the body against foreign invaders. Some T cells regulate immune response. Others bind and kill cells infected by viruses. T cells can protect against reinfection by providing a durable memory of past invaders. And finally, T cells secrete chemicals that help B cells produce antibodies, among other important functions. Anthony, you have studied T cells. this is your backyard here. What does COVID-19 do to our T cells to cause immune dysregulation?
So it stimulates them in a very strong manner. So you can look at T cell phenotypes and kind of gauge where they are on the life cycle. And SARS-CoV-2, or the disease we call COVID, it strongly stimulates a lot of T cells, so much so that we see what's called lymphocytopenia in the acute setting. That means you have low white blood cells, specifically low T cells, because when they're activated, they go out into the periphery, which means outside of your blood. So they strongly stimulate the T cells. When you stimulate cells to grow, they'll age to an extent. There's a paradox with T cells as well. So one would think, with age, you have less activity. But actually, the opposite is true with T cells in terms of their killing activity. So T cells, they must grow up. They encounter what they're going to kill, specifically, usually cells infected with a virus, and then they kill it. And that takes a whole aging process. So the paradox is that when the T cell is older, it's more likely to kill. Okay, so you have young T cells that aren't really killing, then you have older T cells that are killing. So when COVID stimulates the T cells very strongly, the T cells, of course, are hyperactivated. Then they go out and they kill. And also, for some reason, the T cells that kind of regulate our immunity and stop us from having autoimmunity, they change and actually license or actually say, okay, it's okay for you to be an autoimmune cell. So those are called hyperactivated T regulatory cells. So COVID is causing all sorts of T cell, chaos, as it were. So it's absolutely dysregulating T cells.
So you sounded the earliest alarms about this hypothesis and received some pretty bad pushback. And I'm curious, why do you think people were so angry with you, and have they come around to this sort of hypothesis now?
They have, only because, our T cell memory did not bring about herd immunity. So unfortunately, what happened is we studied the T cells in a dish, okay, and said, okay, look, the T cells are responding to viral proteins or antigens, okay? And we said, therefore, our T cell memory should be able to deal with SARS-CoV-2. The unfortunate thing about SARS-CoV-2 is it escapes T cells very, very well. It downregulates the machinery that's involved with an infected cell presenting that it is infected to a T cell. And then there's supposed to be a compensatory mechanism of another cell type, the natural killer cells, but those are unfortunately dysregulated as well. So SARS-CoV-2 can very well escape our cellular immune memory as far as cytotoxic T cells go. And well, they're not memory cells, the NK cells. So I knew that, because of the downregulation of that whole system, that our immune memory that we were building from infections would very likely be insufficient. And because of that, people found it, too bad of a message, and, they didn't want to believe it. And a lot of people, with a little bit of knowledge, went into denial and started attacking me. But I said, there's not going to be a herd immunity to this. The T cells aren't going to help us to this extent that you're thinking. And unfortunately, it's played out that way.
Colin, I'm going to get your take in there. In the last couple of months, support for the idea of that COVID is creating immune dysregulation has become more popular. It seems, I understand with COVID, everything is changing. It is fast, it is moving. As we talked about, the data is delayed, there's not enough. It does seem like there's a bit of this conversation similar to, is COVID being transferred through droplets versus air transmission, and there was this debate happening. And there's a number of debates that are still happening here, and I'm curious, why do you think that's the case in terms of the flack that Anthony was getting about T cells and sort of their impact and not necessarily giving that enough attention?
medical science moves slowly. It's very powerful in knowledge discovery. It gets it wrong sometimes, but it moves slowly. And as you pointed out, COVID is not moving slowly. It's moving very, very rapidly. And that difference in speed creates a lot of tension between those who latch on to new observations and those who remain really yoked to conventional wisdom. That creates also a lot of conflict. It's hard for some people to unlearn. I think some of the most vigorous opponents to the idea that COVID spreads through the air are those whose careers are most invested in maintaining that it's droplet spread. And that's difficult. And this has played itself over and over again in history. But there's one logical thing to point out here. A virus has two strategies, broadly speaking, in terms of being successful. And I think we can agree that COVID is a pretty successful virus. It can mutate to change its appearance to avoid the immune system, and COVID does that. So does flu. Every successful virus seems to be able to do that, or most. But it can also attack the immune system. That's another legitimate strategy. So to say that COVID is not causing immune harm is to say that somehow it's become this amazingly effective virus by only really using one of the two strategies available. Sort of a Darwin approach to this would say, well, survival of the best fit. And so if a virus is able to evolve to harm the immune system, and I think that's what's happening in real time with Omicron. I'm not an immunologist, but that looks plausible to me. I think it's harder to argue that no, it couldn't be or shouldn't be or wouldn't be, because this is how evolution works.
Don, I'm going to come to you. If COVID wears down T cells and does so increasingly with each reinfection, where does that leave us in our fight against the virus?
It makes vaccination all the more important, frankly. And I think, although we may disagree on some of the degree of these various different things, what it does tell us is you need a strong base of vaccination before you get that infection. And the stronger, the more recent your infection is, the more proximal it is to when you just got your last vaccination, the better it is. And so one of the challenges that I think we're also experiencing is the fact that our childhood vaccinations for influenza are terrible. Our childhood uptake for COVID vaccination is terrible. And you need to give your immune system the tools to fight this virus in a way that minimizes the chance that it's going to use those evasive tools and turn your immune system on yourself. And vaccination is our best tool. Masking is our next best tool, because as much as we would like those vaccines for RSV to be in the arms of our kids today, they are not. And so the best thing to do is not get infected. And you can increase the chance of that not happening by wearing a mask. The second best thing is to give your immune system the tools it needs, and that's to have a recent vaccination.
All right, in our last 30 seconds, Anthony, I'm giving you the last word here. Will COVID become the quote-unquote forever virus?
I think it is for now until we get better technology. We don't have the surveillance systems or capacity right now, barring what China does to deal with it. So we're going to need a leap of technology to deal with COVID. It is, pretty much the forever plague.
Colin, Don, Alice, and Anthony, I want to thank you so much for taking your time. Of course, you guys are always doing important work, so thank you so much for taking the time and joining us on the program tonight.
Thanks.
Good night.
Thank you.
As a respiratory illness, COVID-19 is hard on the lungs. For very sick patients, that often meant relying on a ventilator to get through the worst. But even in milder cases, the effects can linger, sometimes for months. Joining us now for more on the line from Kingston, Ontario, Dr. Stephen Archer, professor and head of the Department of Medicine at Queen's University. And Dr. Gerald Evans, attending physician in infectious diseases and internal medicine at Kingston Health Sciences Center and chair of infectious diseases and professor of medicine at Queen's University. Welcome to you both.
Thanks for having us, Jan.
All right, so Gerald, I'm going to start with you. Of course, you guys both have a newly released report, but I want to know exactly what we knew about COVID-19 and lungs prior to that. What did we know? Did we know enough?
Well, we certainly knew that the major serious manifestation of this disease was a pneumonia. And that's because the original virus, the ancestral virus that emerged first in Wuhan, and then ultimately Alpha and Delta variants, tended to cause a severe infection in older individuals, and it was principally a problem with the lungs. So it caused pneumonia, and that resulted in some significant challenges with maintaining your ability to oxygenate your body. And so, as you pointed out, people who had that illness oftentimes required a ventilator to manage them, at least through those parts of the illness.
All right, Stephen, I want to come to you. Of course, this research team encompassed a team across this country. When was the research team assembled? When did this project really get going?
Well, it's interesting. The origin story of this, the universities oddly closed down across Canada. And I think one of the things I'd like to get across is research is an essential service in Canada, and we need to be at the bench. So universities in about March, when the first announcements came out about the pandemic, closed down for in-person use. So we were anxious to get our team back to the bench, as they say. And so in the team, part of it already worked with me in my research lab at Queen's University. And then I talked to Gerald Evans, who's an expert in infectious diseases and virology, and got some advice about how we might turn our cardiac lab that studied hearts to study this disease. And then we brought in Gary Levy from the University of Toronto in April of 2020. And then Shay Colpitz, a virologist here, in June of 2020. And then finally, our colleagues out in Saskatchewan at VITO, which is one of the research centers set up to handle the SARS-CoV-2 virus. We brought them in 2021. So that's the timelines of our teams of 24 people being assembled.
And Stephen, so you mentioned some of the members there. Give us a better idea of the scope. How big was this team, and what are the sort of the institutions that were involved here?
So 24 people at least. 24 people are on the paper that published the discovery, including Dr. Evans. And it's based at Queen's University, but with really important contributions from VITO, which is the Vaccine and Infectious Disease Organization in Saskatoon at the University of Saskatchewan. My colleague there, Dr. Aaron J. Berengy, and then Gary Levy at the University of Toronto. So 3 universities, 24 people.
All right, so Gerald, I'm going to come to you. When we talk about the actual study and the actual coronaviruses we're using, what viruses did we use?
Well, initially, we actually used a coronavirus called mouse hepatitis virus. And many, many years ago, I worked with that in Bob Anderson's lab at University of Calgary when I was a research fellow. And that causes a pneumonia in mice. So it was a model that we could use to look at that. And what the real advantage was is that we didn't have to have a high containment lab to work with mouse hepatitis virus. But ultimately, then it really had to move on to closer models, and then we moved to a human coronavirus called OC43. And then ultimately, as my colleague Dr. Archer mentioned, we were able to do work with SARS-CoV-2 through the VITO organization, which has a high-level containment lab, which allows us to work with that particular pathogen. So depending on how nasty these are to humans, they have to have various levels of containment in laboratories that work with those viruses.
All right, Stephen, I'm going to stick with you. In understanding and better understanding sort of COVID-19 and lungs, I think, as you mentioned, something we need to understand is low blood oxygen. You know, a lot of images of people in hospitals or even at home who have asthma or COPD will use an oximeter to sort of measure those levels. And I'm curious, can you break it down? What is technically low oxygen? What is low blood oxygen?
So doctors call low blood oxygen hypoxemia, which is the fancy word for low blood oxygen. And basically the whole purpose of your lung is to take in oxygen from the environment, and that allows you to make energy in your body. So if you don't have enough oxygen, you can't make the energy molecule ATP, and organs begin to fail. And in coronavirus infections, all 3 of those viruses that Gerald mentioned, they all cause hypoxemia and pneumonia. And what was interesting about the COVID pneumonia from the very beginning is doctors were noticing oxygen levels were lower than they were used to seeing with equivalent pneumonias caused by other bugs. And there was, they quickly found out that this was due in part to the fact, you know, the lung, most people don't recognize this, the lung has its own circulation called the pulmonary circulation, and normally it has an oxygen sensor. So if you have pneumonia in the left lung, it diverts blood to the right lung. but something was happening in COVID pneumonia, that blood was going to areas that were full of pneumonia and thus couldn't take up oxygen. And so patients were coming in and they had two features there. Oxygen level was very low, which might land them on a ventilator. And the other thing is, at least some of these patients weren't as concerned and short of breath as you might expect with a typical pneumonia. So we began to recognize this was somewhat unusual, and we wanted to understand why didn't they sense the oxygen and what was killing the lung cells in about the 5% of people that wound up in trouble with COVID-19.
I'm hoping you can get us some idea of what percentage of people who get COVID suffer from low blood oxygen, Stephen.
And Gerald can weigh in on this too if you want, because he's been our go-to guy throughout this pandemic. But basically, there's about 44 million Canadians have been infected. That's probably a gross underestimate. And about 47,000 people have died. So about 1% of people die. About 5% wind up in hospital, acknowledging that we probably only detect 1 in 5 or so infections. Wouldn't you say, Gerald?
Yeah, those are pretty accurate numbers. And I guess it's also important to point out that during the sort of first year and a half of the pandemic, we were dealing with variants and the original virus, which tended to produce this more profound picture that was studied. Since Omicron's come out, we know that Omicron, that variant and all of its subvariants, tends to cause infection more in the upper respiratory tract than the lung, although we still see lung damage and pneumonia occurring, particularly in elderly individuals. And, that was really the crux of trying to understand it. And I think many people can remember early in the pandemic, there were these really kind of worrisome photographs and film that came out that showed people collapsing in the street with COVID.
Right.
And that was essentially one of the really important things that this research answered, which is, why do people not perceive that their blood oxygen is low to the point that they actually collapsed without sort of having any symptoms? And this was really critical to understanding, because it was a remarkable thing that we saw early on in the pandemic.
Gerald, I want to stick with you. Of course, as you mentioned, there are different demographics here we're dealing with. Some people have healthier lungs than others. Are people with underlying health conditions like asthma, like COPD, more likely to be hospitalized, or is everyone at risk here?
Well, I think the most important thing, and I can't emphasize this enough, age is the critical factor for the severity of COVID. The older you are, the more severe a form of illness you're going to get. Interestingly enough, there have been studies which have looked at asthma, looked at chronic obstructive pulmonary disease, or COPD as it's called, and they've been kind of somewhat contradictory, probably more so with asthma. Asthma patients, surprisingly, at least in large studies that were published back in 2020 and 2021, don't seem to do as badly. Patients with COPD, who oftentimes have more than just airways disease, but structural lung disease, they can do worse. And especially, we know COPD is a disease which tends to occur in older individuals, which again is the primary determinant of more severe illness.
All right, Stephen, before we get deeper into the science, help us understand why it's so important to understand what's happening on a cellular level when it comes to COVID infections.
Well, I think it might be tempting to think that we have vaccines and we have drugs, so why do we need to know any more? We know it's a viral infection. There are several things that I'm sure you're aware of, though, that tell us that we need to perhaps find other tools to treat these infections. First, this is the third pandemic we've had with the coronavirus in just 20 years. So we had SARS, we had MERS, and now we have COVID-19. So this type of virus, which is actually a pretty widespread virus, is going to probably come back. So we need a good understanding of how it actually damages the heart and lungs if we want to combat it. The other thing, too, is we learned a lot about ourselves as human beings, and 20% of our neighbors will not take a vaccine, and worse in other countries. And we also learned that Canada doesn't make drugs, so we're sort of downstream from the largesse of other countries in terms of getting these things. So we need some better understanding of the virus so we can prevent its ravages in the people that are most susceptible. And we need to recognize that there's not going to be just one tool to attack to prevent damage from the virus. And furthermore, the stuff that we discovered in this paper, interestingly, seems to apply to most coronaviruses, including prior viruses like the virus that caused SARS. So there seems to be some commonality in this mitochondrial mechanism that we have uncovered.
Well, I'm going to stick with you and let's talk a little bit about that. And I want to make sure that we get, what was the central goal of your research when we talk about not only the pneumonia aspect, but also on the cellular level?
So we wanted to discover two things with our research. One is, why was the lung so whited out on the chest x-ray? What caused that lung injury, which is called diffuse alveolar damage? And the second is, why was the oxygen level so disproportionately low in the lungs? Those were our two goals.
Gerald, I'm going to come to you. You touched on this a little bit, but what distinguishes COVID patients with pneumonia from non-COVID patients with pneumonia?
Well, principally, if you look at adults, most pneumonias are actually bacterial. They're caused by bacteria, not viruses. So it's not that viruses can't cause pneumonia in adults. We know that influenza does, for instance. And in some people, even viruses like RSV and rhinoviruses, which cause the common cold, can cause pneumonia. But what's unique about viral pneumonias in general is that they tend to affect all of the lung. Bacterial pneumonias oftentimes may be sequestered in just one part of the lung, what we call a lobe. So a lobar pneumonia is very characteristic of bacterial pneumonias in adults. And the other thing, of course, is that the degree of hypoxemia, the degree of oxygen loss, although it's seen commonly across most pneumonias, it's very, very severe with some viruses like influenza and like what we're seeing with the coronaviruses in general. So our understanding of how that happens and is really critical to our developing therapeutics and also understanding what ultimately is happening in the lung over the long term, because as you commented on early in the preamble, is that, you know, the effects of COVID after they've infected someone may persist for a while, and some of that is going to be due to damage in the lung, and understanding that is absolutely critical. And that's what the real key to this study has shown. And I would say it really underscores why we have to understand how viruses cause disease, or what we call viral pathogenesis, because that's a fundamental piece of understanding we need to have in order to move forward with ability to develop therapeutics, for instance, and figure out, you know, how do we as physicians treat patients who have this infection.
Stephen, I'm hoping you can break this down for me. Someone who has COVID-19 or is recovering from COVID-19 is having issues breathing. What does that look like when they are taking, you know, some air and oxygen into their lungs, and how does that sort of work in terms of their lungs and where the oxygen is actually going and getting oxygenated?
Well, if you start with like what we know about the pathology of this disease, in other words, what the tissues look like, their lungs are partially full of liquid. So oxygen doesn't go through that liquid very well. They're also a little bit scarred or fibrotic, so their lungs are stiff. And so when you combine all, and they're very inflamed, lots of white cells of various types of immune cells have infiltrated their lungs. So every breath they take is much more work than it should be. And whether they sense that work or not, they eventually get tired. And what starts happening is their oxygen level goes down. They become what we call cyanotic, which means purple like a grape. And at some point, if they don't get assistance, they will stop making energy and they'll get heart disease, lung disease, and they'll wind up on a ventilator. I should mention this virus doesn't just infect the lungs, although that's its number one target. It's perfectly happy to bother your heart. It can affect your kidneys. Your intestines are a big site of coronavirus infection. So all these organs begin to fail. So the person is not only hypoxemic, but they're suffering from various other forms of organ failure that can threaten their life, and both acutely and then in the longer term. And I should mention also, and maybe we'll get to this in the basic science, Jam, but the viruses have a long history of not liking bacteria. They fight each other. There's a long-standing war between viruses and bacteria, and our mitochondria inside our cells once were bacteria. So we're sort of bystanders in this war between viruses and bacteria, with our mitochondria being to some extent mistaken as bacteria.
So we'll touch on the history, but I want to stick with you in terms of how the coronaviruses damage mitochondria. What does that actually look like?
So I like to think of it like alien versus predator. These two nasties are fighting with each other. The virus comes in and it got RNA as its code, and it basically takes over our cells and uses us to make a sort of like a photocopier to make copies of itself. But an interesting thing it does, and I don't know whether the virus intended this or not, but its code basically turns on a lot of our genes or turns off a lot of our genes, which target our mitochondria and our lung cells. And it also makes proteins which are part of itself. It doesn't have very many proteins, but these fundamental building blocks, some of them go right to our mitochondria and other critical structures in our cells, and they engage in what's called protein-protein interactions, and they often block the normal function of the cell. So they use our nucleus, our genetic code, against us, and they also make their own viral proteins, which are toxic to our cellular proteins.
And Gerald, I'm going to come to you in terms of adding onto that. Were these the findings that you sort of discovered? Have we seen this in other sort of, not only coronaviruses, but other viruses in terms of how the mitochondria sort of react to these viruses?
Yeah, no, it's very fascinating because when Stephen and I first started our discussions about this long ago in the early part of the pandemic, actually I found a few papers that discussed the role of mitochondriopathy or disease in the mitochondria and in other viruses, particularly coronaviruses, but other viruses as well. But sort of the really fundamental understanding of how that happens is really part of what this study was all about. And I think this study has really expanded that area to make us understand how important that kind of interaction is from other viruses and how it very severely affects cells. If cells can't make energy, as Stephen was mentioning, they're going to die. And one of the things that's really interesting, again, about the studies that Dr. Archer's done for many years with mitochondria is understanding how dysfunction and damage to the mitochondria eventually results in cell death, or what's called apoptosis. And that is fundamental to us understanding reversibility or irreversibility oftentimes of various virus infections.
All right, Stephen, I want to come to you. You talked about alien versus predator here, and I want to get through some of this history. There's an interesting, as you mentioned, evolutionary angle to this. Talk to us a little bit about how viruses, bacteria, and mitochondria are all sort of linked in this when we talk about COVID-19.
So I'll go back to 1967, which was not only Expo in Canada, but it was also the year Lynn Margulis wrote a paper basically saying that once long ago in the primordial soup, a bacteria that could actually use oxygen to make energy got taken up by what eventually became us, human cells. And so now think of a human cell with these former bacteria that know how to make energy inside them. But what's been going on for almost as long as that is viruses have been attacking bacteria. And what happens is the virus inserts its genetic material into the bacteria, and then the bacteria fights back with its own immune system. If you can believe it or not, the bacteria have an immune system. And in fact, that system, which is called the CRISPR-Cas system, CRISPR-Cas chops up the viral material and protects the bacteria. Human scientists recently used that discovery of the CRISPR-Cas system and made a Nobel Prize-winning discovery about how to edit genes and cut genetic material to fit your needs. But that's a pathway long ago in bacteria. So we're now set up that when this virus comes in, and SARS-CoV did the same thing, we can look with big genetic screening programs and just say, when you get an infection, what happens to your cells 2 hours, 12 hours, 24 hours after infection? And what you're seeing is this virus is basically turning thousands of our genes on and off, and it's making proteins which are toxic to the cell.
Gerald, I want to come to you in terms of how we can use this research not only for other viruses, but with long COVID. That is a big player here when people are infected and they're suffering not for months, if not years. Can this be applied?
Absolutely. And I think this is one of the critical things. So as many people know, we're trying to understand what is it that drives long COVID. As an infectious disease doctor, I've known for many, many years that various infections leave people with prolonged, persistent symptoms that can go on for months and months, if not in some cases years. And so trying to understand what is the mechanism by which that infection causes that problem is really important. So there's lots of speculation. Is it persistent virus? Is it the immune response, which is sort of continuing on long after you've recovered from the illness? What I thought was very critical, and this is speculative, is that if the viruses are causing significant damage to mitochondria, even if that's a reversible process, that mitochondria is the power supply of the cell, as Dr. Archer will tell you very many times. That's critical to how cells maintain their health. And healthy cells are what drive all of these systems in our body that allow us to, to digest food, to think with our brain, to have the heart pump and stuff. So if this mitochondriopathy, this disease of mitochondria, which is caused by the virus, is significant and lasts for a considerable amount of time because of, say, viral persistence, then that will very much help us understand what causes long COVID in particular, and potentially at least what causes a lot of these persistent long-term symptoms that have occurred in people who've been infected with other viruses as well. So I think this actually very, very much excites me into understanding that. And again, I can't emphasize how important the work of scientists like my colleague Stephen Archer is. Understanding how viruses cause disease is fundamental to us being able to understand what those processes are and what we can do as physicians to develop treatments and or understand that whole process a lot better. It's incredibly important.
Stephen, you get the last word on this. Now that we have this information, that you guys have put several months of work into this, how can we better prepare for the future?
Well, for our modest discovery, I think the thing is we show the virus has broken the mitochondria into pieces. We're developing drugs to prevent the fragmentation of mitochondria. We've shown it turns on apoptosis. We're developing drugs, small molecules that attack that. But the one thing I would be remiss if I didn't comment on is the incredible courage of my colleagues, the coworkers who are at the bench. And these 24 scientists, right now, we now have vaccines. Everyone feels a little bit more comfortable. But in 2020, people were afraid. We didn't know, could MHV1 infect humans? We didn't know how safe it was to work with HCOV-OC43. And these people came in when most people were forced to stay home. They were at the bench every day working on this. And they came from India and Iran and England and all over the world, Brazil. And they were so passionate. They are so passionate about their discovery. And I think a couple of things that I'd like to just leave Canadians with listening to this is we need to better fund research in Canada, because these people live tenuous lives. They live from grant to grant. If I don't get research funding, they don't have a job. And we need more continuity in science and the ability to go deep and try to invent something. That's certainly something. We invented insulin here in Canada, or discovered it, at the University of Toronto. We can certainly do that for new therapies for COVID-19. So that's the main thing I would leave your listeners with, Jan, is just I think we've discovered that mitochondria are targets of virus, and we can develop drugs to stop that. Made in Canada science is important, and we bring the best people from around the world because they're passionate about having a chance to do science and work with docs like Gerald, and so I think we should do more of that.
Stephen, Gerald, thank you so much for the work that you do, and thank you so much for joining us on the program tonight.
Thank you for the time. Nice talking to you.
That is the agenda for Monday, November 14th, 2022. Tomorrow, Steve finds out how our brains make memories and why being unreliable might actually be part of how they're supposed to work. I'm Jay Jaganathan. Thanks for watching TVO, for joining us online at tvo.org, and Steve, we'll see you tomorrow.
The Agenda with Steve Paikin is made possible through generous philanthropic contributions from viewers like you. Thank you for supporting TVO's journalism.